And so my talk is going to be titled Toepad Shape, Allometry, and Evolution Across Hemidactylus Geckos. And so this is a talk for the Society of Integrative and Comparative Biology Conference 2023. My name is Travis Hagey. I'm a professor at Mississippi University for Women. And this is a project that was done by one of my students, Alaina Dawkins, who's graduated now, undergraduate student. So I want to start off with some acknowledgments. Again, was Alana's project that she did in my lab. And so as part of this project, she was able to get funding from the Mississippi State Space Grant Consortium and our own in-house Mississippi University for Women Summer Scholars Program. And so this was a project she worked on for one whole semester and then a summer. And so everyone in my lab, we study gecko lizards, and they're a big, diverse group of lizards that are really fun to study. And more specifically, we study their toepads, which are kind of well known for having sticky feet. And so the toepads probably evolved multiple times within geckos, and they were probably also lost multiple times within geckos. And we know this because of some of their internal anatomy. We can tell which groups probably never had toepads, which groups probably had toepads and may have secondarily lost them based on their internal toe anatomy. And we can also have evidence from ancestral state reconstructions that there were probably multiple gains and losses. But pinning down exactly where in the phylogeny those gains and losses were can be kind of difficult. And so in this study, I want to talk about this origin right here. And I know you can't really read any names on these phylogenies, but this origin is at the base of a genus of geckos called Hemidactylus. And the reason we think there was probably an origin at the base of Hemidactylus is because all the species within Hemidactylus have toepads, but a couple of the sister genera and other closely related groups near Hemidactylus, none of them have toepads, or at least for the most part, none of them have toepads. We'll talk about Cerdidactylus at the end, but a lot of the closely related groups are padless or nearly padless, which suggests that there was an independent origin of toepads at the foundation, at the base of Hemidactylus, which is the group we're going to be talking about in this talk. And so why should we study the genus of Hemidactylus geckos at all anyways? Well, they're one of the bigger gecko genera. There's almost 200 currently described species. They're a relatively old genus of geckos. Geckos as a group are probably 200 million years old. Hemidactylus as a genus is probably a little less than 100 million years old. So they're an old, big group of geckos, and we can find them around the world. Pretty much anywhere that's tropical, temperate, or arid environments, there's probably Hemidactylus species there. And so they, on their own, over the history of the clade, they've been really good at colonizing new environments. And then even more recently, humans have also been really good at spreading them around. And so if you are kind of familiar with geckos or doing tropical herp research, you're probably familiar with some of these species. So Hemidactylus tursicus, the Mediterranean house gecko, has been introduced to a lot of different places, including the southeastern US. We have them here in Mississippi. And Hemidactylus frenatus, the Asian house gecko, which is a little bit more tropical than Hemidactylus tursicus. We have them in Florida, we have them in Hawaii, and a whole bunch of other places around the world. But these aren't the only two Hemidactylus that have been successful colonizers. They're just kind of the most two common. But Hawaii has other species of Hemidactylus. I want to say Louisiana has a third species of Hemidactylus. So these aren't the only successful invasive species in Hemidactylus, but they're maybe the two most well known. And so as far as the toepads within Hemidactylus, most of the species in this genus have kind of a stereotypical toepad that's comprised of multiple pairs of lamellae or ventral scales on the bottom of their feet that have adhesive structures. And so this is a pretty common morphology in Hemidactylus, having these sets of pairs of toepads or adhesive structures, toepads. But there is some interesting variation within the group. And so this is, we're using kind of Hemidactylus tursicus as kind of our stereotypical Hemidactylus. But there are other Hemidactylus species that have much bigger, exaggerated, dilated toepads with many, many pairs of lamellae. Some of them are webbed, some of them are not webbed. These are all pictures of back feet, by the way. And so some species have much more exaggerated dilated topads. On the flip side, some species have really tiny, reduced, narrow topads, but still having ventral scales in pairs. What's interesting is not all Hemidactylus species have paired lamellae. Hemidactylus garnodii is kind of a weird intermediate case where the distal end of the toe has a division in the middle, so pairs of pads, and then about halfway down, that subdivision in the middle goes away, and the proximal end of their toe just has bands of lamellae, so they're not divided into half anymore. Um, an even more extreme example of this is Hemidactylus and amelensis, which is the earliest branch in Hemidactylus. It's one of the oldest lineages within Hemidactylus. Some studies lump them, put them into their own genus, but we're going to leave them part of Hemidactylus just for this study. But instead of having pairs, they don't have any subdivisions. So they have just undivided lamellae, which is kind of exciting because maybe they represent kind of the intermediate or an ancestral state of what Hemidactylus topads look like when they first were developed, before they developed the subdivision down the middle that most Hemidactylus have now. So what was the goal of our study? Was really just to investigate and quantify topad shape variation within the genus, across the genus. And so to do that, we used geometric morphometrics. So we took pictures of the rear feet of as many species as we could get. Some of them were preserved specimens, some were from live specimens, some were right back feet, some were left back feet. If they were a right back foot, we horizontally flipped those images so all the images looked like left back feet. We focused on the fourth toe, which is usually the biggest or kind of most well-developed toe. And we used a handful of landmarks to kind of capture the toe shape as well as the toe pad shape and its location within the toe. So we had 2 landmarks at the base of the toe where the toe meets the palm on the left and right sides. And we had 1 landmark at the base of the claw. And then we had 4 landmarks that captured the adhesive part of the toe. 1 at the proximal end, 2 at the widest points, and 1 at the distal end of the adhesive area of the toe. Then we used curves to connect landmark 4 and 5, 5 and 6, 4 and 5, 5 and 7, 7 and 6, and 6 back to 4 to, in essence, outline the adhesive part of the toe. Each of those curves was comprised of 13 semi-landmarks. So we use this set of landmarks to quantify topad shape and toe shape across all the species we were focused on. And we had 1 image per species. We also gathered some other data. We counted the number of lamellae or lamellae pairs each species had. We categorized each species as divided or undivided, and we used that enclosed area from our landmarks to actually measure topad area. So each of our images had a scale bar, so then we could quantify area that was enclosed by our landmarks and curves. So Reptile Database currently has 187 described Hemidactylus species. We wanted to do some evolutionary analyses, so we needed a phylogeny. So we went to the Tonini squamate megaphylogeny, and it contains 78 species of Hemidactylus. Of those, we were able to get topad pictures for 46 species. 4 of the species we had topad pictures for weren't in the phylogeny. So if we were doing non-phylogenetic analyses, we included those 4 species. If we were doing anything with a phylogeny, we excluded them. So our data set has 46 species in it. Here is the Tonini phylogeny that's been trimmed down to just have Hemidactylus. Names in black were species we had topad pictures for. Names in gray were species we didn't have data for. So you can see we have pretty good sampling across the Hemidactylus phylogeny. To put in context some of the morphological variation in the group, again, species that with black names have kind of your stereotypical pairs of pads along the toe. Species outlined in red have those undivided pads, those where the lamellae are just bands on the bottom of their feet. So here you can see there's that really early species of Hemidactylus with a fully undivided pad. Here's Hemidactylus garnodii, which is kind of half divided, half undivided, and some other species that have undivided lamellae. And then we also highlighted species that have narrower pads. They're still divided, but they're really reduced. And so you can see we have a couple of them here. So you can see morphology is kind of variable across the phylogeny. These weren't changes that happened just once. So there's some interesting variation across the group. So we were just interested in quantifying that. So this was an undergrad project, and so we spent a lot of time going through checking data quality. So we spent a lot of time plotting and evaluating, making sure all of our landmarks were in the right places, all of our curves were connecting the right landmarks, that they were put down in the right order, and they had the right number of semi-landmarks. We went through and kind of methodically compared each landmark against that same landmark in other specimens to make sure, again, that none of our landmarks were in weird places or places they shouldn't be. And since we were using the included scale bars in our images to calculate area, we double and triple checked that our scale measurements were correct too. So the first kind of set of analysis we did was just a principal component analysis, just to look at the variation in our data set. And so you can see each specimen or each species is a dot here, principal component 1, 2, 3, and 4. So by the time we get to PC4, we're capturing 70% of the data. So we're capturing a lot of the information here. You can see the dots on our plots here are colored by the number of lamellae each species had. So some species had as few as 7 lamellae or lamellae pairs, and some species had all the way up to 16. So there's some variation in how many lamellae species have. You can see principal component 1, in essence, captures that concept of kind of a skinny, narrow toe or a big, fat, dilated toepad. Principal component 2 starts to capture how much of your toe is covered in toepad. Is the adhesive part of the toe kind of towards the tip of the toe, or is the whole toe covered in toepad, and does the toepad actually extend down into your palm? What's interesting here too, if you kind of squint, you can see a lot of these warmer colors tend to be up high, larger values on PC2, whereas lower values on PC2 tend to have these cooler colors. So kind of squinting at this, it kind of suggests the more of your toepad that's covered, more of your toe that's covered in toepad, the more lamellae you might have. And that's something we go on to test later on. By the time we get to principal component 3, we've captured 50% of the data. And so we start to get into kind of some weird asymmetries that was present in our data set. So kind of how straight the sample was, or if the claw got pushed over to one side while we were imaging it, as well as kind of how exaggerated or how dilated the toepad is, right? How big the toe is. Same thing for PC4. We're getting kind of some weird asymmetries, but also how far down into the palm the toepad goes. The toepad goes. So when I first saw this, the first thing that jumped out at me was this point right here. The lines connecting all the dots are the phylogeny. So you can see this dot isn't connected, meaning it's one of those 4 species that wasn't in the phylogeny. And so this is Hemidactylus richardsoni. So when I first saw this, I was thinking maybe this specimen has a landmark that's out of place and making it seem really weird. But after looking at it closer, I don't think so. This is kind of has some webbing between the toes, which makes it hard to place these landmarks 1 and 2. So it's kind of why we have some weird asymmetry there. And you can see this claw kind of got pushed over to the side. Not that the specimen is really shaped that way. It's just the way the specimen was laid out when we took this image, which again is kind of this asymmetry, which is more a methodological thing than actually a real biology. So I think that's a real location of our landmark. I don't think that's an error. I think we're just getting down to the point by PC3, we're capturing just noise in the data set. So then we wanted to do some kind of basic analyses just to look at some of the basic patterns in our data set. So the first thing we did was we wanted to compare body size, snout vent length, against toepad area. This has been done in a lot of different species of lizards with toepads. We had a pretty solid expectation that bigger animals tend to have larger toepads. We did this using phylogenetic generalized least squares, where we estimated Pegel's lambda. Clearly, there's a lot of phylogenetic signal, but also bigger species tend to have bigger toepads. That's not surprising. The gray lines in the back there are the phylogeny. And again, a similar analysis, we did body size, snout vent length, against the number of lamellae each species has. Again, we found a significant positive slope. Bigger animals tend to have more lamellae. But what was interesting was the slope of these two analyses were really different. And it didn't seem like area and lamellae were all that closely connected. So we went further, we did a third analysis where instead of using Toepad area, we wanted to measure Toepad length. So we went back to our original landmarks and calculated the distance in millimeters from landmark 7, which was the distal end of our Toepad, to landmark 4, which is the proximal end of our Toepad. So that's just a straight line measurement of how long the Toepad is. So that was Toepad length. We compared that to lamellae count, which seemed like a better comparison as opposed to comparing area and count. So when we looked at length and count, we found a really linear relationship, a positive relationship, significant relationship. And so what's interesting is the slope of this line is 1.18. Remember, slopes is rise over run. So for every 1.18 lamellae a species had, their Toepad was 1 millimeter longer. If you divide that out, it suggests that maybe lamellae size is standard across species, regardless of how big they are. Maybe all species generally have lamellae that are about 0.85 millimeters wide. And so this would be a really cool future study to do, is to explicitly measure how thick, how wide, or tall each lamellae is, and see how universal this width is. Because that would be really interesting if lamellae are the same size, regardless of if they're on big animals or small animals. That would have really cool kind of biomechanical implications for how topads work. So those were kind of some cool analyses, some cool results. We also wanted to look at how topad shape correlated with things. So we wanted to ask, okay, species that have more area, are they shape different? And again, we did this using phylogenetic generalized least squares. And after accounting for body size, then we looked at the remaining variation and how we could describe that with shape, how we could correlate that with shape. And so we saw animals with bigger topads also had significantly differently shaped topads. And so you can see animals with bigger pads tended to have these fatter, wider pads, whereas animals that had smaller topad areas had narrower topads, which seems intuitive. We also did that with lamellae count. Again, after controlling for body size, species with more lamellae also were shape different, significantly shape different. And again, it's a really subtle difference, but you can see species that have more lamellae have topads that extend a little bit closer into the palm as opposed to species that have fewer lamellae, which again backs up the analysis we just did earlier, where species with longer topads tend to have more lamellae. So all of this is making sense, and it's pretty intuitive, which is really cool. We also wanted to ask if species that have divided pads versus undivided pads, ask if they're shaped different. The tough part about this analysis is we only had 3 species with undivided pads, so we didn't have a ton of statistical power. So this analysis was not significant, but when we did a CVA, a canonical variance analysis, so which finds an axis that most closely captures the difference between our categories, divided and undivided, we do see a pretty stark difference. Not that you can totally take this as meaning significant differences, because we didn't have the power for the differences to be significant, but these results suggest species with undivided pads tend to have these narrower pads, which intuitively makes sense, versus species with divided pads tend to have fatter, wider pads. So that was kind of some of our just basic understanding the variation in the group kind of analyses. The next thing we wanted to do is take a more evolutionary approach. And so we did something called a node height test. So a node height test relies on calculating standardized phylogenetic independent contrasts. So traditionally, phylogenetic independent contrasts use univariate measurements where you calculate the difference between some tips or difference between nodes. If you know the branch lengths, you can in essence calculate how much trait evolution had to have occurred on these branches to result in the differences you're seeing in the taxa, which is in essence an estimate of the rate of trait evolution. And you can do this for each node going backwards through time, as long as you can calculate weighted averages of your trait for each node going back in time. So what we did, instead of using univariate variables, we were able to calculate phylogenetic independent contrasts using shape, using our multidimensional shape data. So instead of just differences in the traits, in trait values, we calculated across these different distances to estimate how different each shape was. We had branch lengths from our phylogeny, and we were able to calculate weighted shape averages for each node going back in time. So now we were able to, using this approach, we were able to estimate the rate of shape evolution for Toepad shape. And so then once you have an estimate of rate for each node going back through the phylogeny, you can see how that rate of change changes through time to evaluate different models of trait evolution. So here is our Hemidactylus phylogeny with all the species we had data for. Here is, on the x-axis, we have time, millions of years above the root, and our standardized phylogenetic independent contrast of shape, which is in essence an estimate of trait evolution. So hypothesis-wise, if your standardized independent contrasts are relatively constant through time, they're the same in nodes that are earlier in the tree versus later in the tree, that suggests your rate of evolution has been constant through time, which is consistent with Brownian motion. Alternatively, you might see really fast rates of trait evolution here earlier, deeper in the tree, and slower rates more recently in the tree. That would suggest an early burst pattern of evolution, where evolution was really fast earlier in the phylogeny and slowed through time. There are lots of examples of this in previous studies that suggest maybe some sort of niche filling model or ecological opportunity that's being filled up as the clay diversifies. Lastly, something that's not seen very often is a late burst model of evolution, where evolution was slower at the deeper nodes and faster more recently. Interestingly, for this data set, we saw a late burst model of evolution, or at least our data is consistent with a late burst model of evolution, whereas we found really slow rates of shape evolution early in Hemidactylus and much faster rates later, more recently in Hemidactylus, which is kind of a really weird pattern. I'm not really sure quite what to make of it. Some people have suggested that maybe this is some sort of multi-optima OU type situation where clades or taxa are still moving towards their optima, multiple optima. So if we look at shape, we found a late burst model of evolution. If we redo those same analyses, but we do it with Toepad area, we see a constant rate of change, suggesting Brownian motion. If we do the same thing with lamellae number, we see a constant rate, suggesting Brownian motion. So this is even more interesting. Why would Toepad shape be a late burst model of evolution, whereas area and lamellae number are Brownian motion? Super cool. I'm not really sure how to interpret these results, but they're really neat. I'm really excited about them. So what could this mean? Well, again, Hemidactylus, depending on how you define it, are probably a little less than 100 million years old. They can trace their ancestry back to Asia. And so what was happening around that time geologically was that's when India was crashing into the Asian continent. And so maybe this is related somehow. Were Hemidactylus on the Indian subcontinent, and when it crashed into Asia, allowed them to colonize the rest of the world? Maybe, but the sister groups to Hemidactylus were also from Asia. So that doesn't quite make sense. Where if Hemidactylus were already present in Asia, maybe India crashing into Asia changed something or promoted the origin of Hemidactylus as a group in general. And then once Hemidactylus appeared, then they started colonizing the rest of the world. They colonized the Mediterranean, they colonized Africa, they jumped over to South America, they colonized all the way down into Southeast Asia, into Australia, and out into the Pacific Ocean. So they already have a really strong track record of colonizing new environments. And so I'm wondering if maybe them colonizing new environments generated faster and faster rates of evolution, right? The individuals that could adapt to new environments quickly were the successful groups, which promoted them to colonize new environments even faster. So maybe they've already kind of naturally evolved over the last 50 million years to be these quickly evolving, hyper adaptable group. Um, and so to kind of back this up, I'd love to see somebody do kind of a paleobiogeography study of Hemidactylus and Dravid gecko, the other genus that early clade is sometimes put into, um, to see if we can kind of find some kind of biogeographical explanations for why their rate of evolution might have increased through time. If that gave them a kind of an edge colonizing new continents, that might be why. Another really cool future study I'd like to see is a similar kind of analysis looking at toe and toepad shape in Cerdidactylus. So Cerdidactylus is the sister genera to Hemidactylus. Most Cerdidactylus don't have toepads. And the origin of Cerdidactylus probably didn't have toepads. But Cerdidactylus you can find throughout Asia, Southeast Asia, and Australia. Some of the Southeast Asian and Australian groups do have rudimentary toepads. So it looks like there was an origin of toepads within Cerdidactylus, not at the tip, not at the root of Cerdidactylus, but within Cerdidactylus. So I'd love to see this same kind of study quantifying toe and toepad shape in Cerdidactylus to see if they also follow a late burst model of evolution, since they contain an origin. And so if you think this stuff is interesting, let me know. I'd love to hear your ideas about late burst models of evolution. But if you think this kind of stuff is interesting, definitely check out my other student's talk, Jenny McCann's. She's going to be talking about toepad shape across all geckos, across all 6 families of pad-bearing geckos. And so she gave her talk at SICB, but her talk is also going to be here on SICB+. So let me know if you have any questions. Thanks.